It's a huge pleasure to be here to talk about what's new in palm systematics and evolution. Thanks to a suggestion from my colleague Lauren Gardner, I retitled this slightly. This isn't supposed to look like Genera palmarum burning up in a black hole, more Genera palmarum heading into a large-scale phylogenomic future, which is, I think, where we're heading. So in 1997, I attended the first palm meeting I ever attended, which was a nice, cozy, invitation-only meeting at New York Botanical, where I met quite a few of the key, almost all the key palm figures, including many people who are here today. And at that meeting, Natalie Yule gave a really nice, reflective talk on Genera palmarum edition 1. What it had done, and also, I think, importantly, what it hadn't done, what it needed to do, what people needed to do, responding to the content of GP1. We've seen some of these, there's a little bit of repetition here at the beginning, but just to put GP in context, in case there are a few people who weren't here yesterday, the Genera palmarum project grew out of foundations laid by Harold E. Moore Jr., a truly legendary palm researcher who had always had an ambition to pull together a complete generic monograph of the palms. But sadly, he died unexpectedly in October 1980, and he left behind some very important contributions, such as this, his most important paper, the major groups of palms and their distribution, but he had not written Genera palmarum. And this task was left to John Dransfield and Natalie Yule, as John explained yesterday. Genera palmarum edition 1 was published in 1987 and was the first modern systematic treatment of palms, and it recognized 200 genera at the time. I think we, as John was talking yesterday, I realize how we almost take for granted our state of systematic knowledge of palms now, and the mess that John and Natalie untangled and synthesized in that book is truly extraordinary. John mentioned incremental improvements. The very most significant increment that was made took place when that book came out. And really, it was an incredibly important springboard for new research, and you can see that by looking at its citation stats. There are approaching 800 citations for GP1 on Google Scholar. And again, we've seen this picture yesterday. Through the 1990s, John and Natalie started having summits in the Montgomery Botanical Center, and young Turks and whippersnappers started participating, and it became clear that a broader-based authorship is going to be necessary. And here you can see all the authors of Genera palmarum edition 2. There's me, Connie Asmussen, Natalie Yule, John Dransfield, Madeline Harley, who didn't make it to that meeting, and Carl Lewis. And Yatney there, ever present to make sure that we were sufficiently fueled with excellent foods to get through our discussions. GP2 came out in 2008, and it really felt like giving birth. Should I actually know what that feels like? And it was a major punctuation point in the development of palm systematic research. This treatment recognized rather fewer genera, 183, than the original first edition. I did a bit of a back of envelope calculation. This is a much heavier book, and it contains roughly 40% more content than the first edition. That's both in terms of words, pages, illustrations, and so on. There are major additions in GP2 that were not featured so conspicuously in GP1. A lot of phylogenetic data, a much deeper biogeographic discussion, a much more extensive synthesis of fossil and pollen information, many more colour images, more maps, and a significantly expanded glossary. It's a bigger and better book in many ways, but both books still play a role. There are still things in GP1 that are not in GP2. And all of this content was structured around a phylogenetic linear classification. What is a phylogenetic classification? Well, we aimed to nut out a classification that was consistent with phylogenetic knowledge at the time. And we did that at a summit meeting in Cornell, where we sat around a table in the Bailey Hortorium, which was then outhoused while the hortorium was being refurbished in a curious factory. So it was a strange industrial setting for the devising of a phylogenetic palm classification. We sat there all week with printouts of papers and unpublished data and attempted to bring together a linear classification that reflected phylogeny. Now, phylogenies are not linear. They're not intended to be linear, and there are many, many ways of rendering a branching diagram into a linear sequence, so you have to apply a set of rules. So in short, what we did was building on the best phylogenetic evidence for a particular group. We worked from the base of the trees, node by node, picking off the smallest sister taxa in turn. So you can see in this diagram here, I've tried to draw it to understand it. Working from the bottom, we would work up this branch here with fewer taxa on it. It's equivocal whether these are first or second here. That could have been switched the other way around. And then to the larger clades here, where we would pick off the smaller lineage and work inwards. This is not to imply any kind of linear sequence of primitivity, primitive to derived in the sequence itself. The aim is to make sure that closely related taxa are close to each other in the classification. This classification was then published in Kew Bulletin in 2005, and also another remarkably highly cited paper, given that it's a rather dry validation of this new classification. Okay, so what's changed? What's changed since GP2 and now? Well, GP2 recognized these numbers of subfamilies, 5 subfamilies, 28 tribes, 27 subtribes, 183 genera, 17 unplaced genera, and around 2,400 species were enumerated in there. The figures today actually look rather similar, and the variation can be seen here in genera. There's one less genus, and there are rather more species. I'm going to skate over species taxonomy because that wasn't the main aim of genera palmari, but I do want to draw attention to some interesting stats here. Since GP2 was published, there have been over 200 new species of palm described. And if you look at the total universe of new names, there are actually over 300 new names, because that not only includes the new species, but also the new combinations and the new names that come about when species are moved between genera. These new names occur in a wide range of taxa that I've just enumerated here, the genera in which there has perhaps been most taxonomic attention and dynamism. And here you see a selection of new taxa that Kew people have been associated with. That's Calamus comsarii, Heterospapi Barfodii, and the beautiful Dipsis McKeary. And just to show you some things I'm quite proud of, we've been cranking out rattans. We've been studying the rattans of New Guinea for rather too long, longer than I dare to mention. Last year we published 15 new species of calamus, and we have another 10 to deliver this year as well. We're very lucky in Kew to have the most fantastic, experienced palm artist, Lucy Smith, who has the ability to take very complex material and turn them into highly informative but also extremely elegant plates. What about the generic taxonomy? Well, I showed you in that earlier table that there's a very small difference in the numbers of genera that are accepted in palms between now and GP2. but in fact there's been quite a lot of turnover. So we have 6 new genera and 7 that have been sunk. And I'm going to go through most of these in brief now. So the first couple of slides are about slide trachycarpy, which Christine Bacon took on for her PhD and moved on very significantly. The trachycarpy, one of the major tribes of the family plant subfamily Coryphoideae, really was problematic for us in GP2, and we were lacking critical information, and Christine's PhD thesis really turned that around. One of the most amazing findings, John alluded to this yesterday, was that the genus Livistona, which those of us who work in the old world know and love, in fact is broken into 2 unrelated clades, and this led to a new genus being required, except that this genus would include a species that already had a genus name that we could use, the genus Sarabus, and in turn we had to sink the New Caledonian endemic Brichardiopsis into Sarabus. So this is a really distinctive palm, in fact. Now we know it, and it has good synapomorphisms such as these peculiar inflorescences which have a basal iteration that within a single profil you have usually three equal axes, equal peduncles, and they don't each have their own branch, so it's kind of anatomically anomalous. So these trifid inflorescences are a key character, and also fruit colour features and anatomical features clearly differentiated from Livistona. And I guess this is perhaps an even more shocking discovery, also made as a result of Christine's work, the genus Lanonia. Lanonia looks, for all the world, like Liquala. Liquala is a major genus of the Asian tropics and has a highly distinctive leaf that is deeply split into a wedge-shaped segment. So the fact that there could be another unrelated clade, more or less unrelated clade, that does this apparently unique Liquala leaf was a major shock. But there are also plenty of characters in the way that the central segment is split, and a very kind of obscure character that at the base of the central segment split around here, you have a tiny little blip, a pulvinus, which you don't find elsewhere in Liquala. And there are also reproductive characters. These plants tend to be dioecious, and there are floral synapomorphisms. Perhaps more remarkable was that although a number of the taxa were already described, Lanonia really seemed to come to light because of all the new Lanonias that Andrew Henderson was discovering on his fieldwork in Indochina. So a really astonishing finding, a genus that appeared from nowhere with many new taxa. I think it's 9 species overall. And of course, I can't come to a meeting in Colombia and not mention Sabineria, undoubtedly the most stunningly beautiful of all the new genera that have been described post-GP2. This belongs to the tribe Chrysophyllae. It's related to Italia, but distinct from it, and occurs up near the Colombia-Panama border, and I'm looking forward very much to being introduced to this palm in person next week. It has a number of really good floral characters, morphological characters. First and foremost, this spectacular leaf, but also a number of floral characters and other inflorescence characters you can see there. My own dabblings in new genera are concentrated in the very slippery subtribe Typhospermatidae. This is a really well-defined arecoid subtribe, which has pre-morse leaflets. These leaflets are very jagged. It has staminate buds that are bullet-shaped. They're always multi-staminate, many stamens, sometimes over 100. And in the staminate flower, you always find this lageniform, this flask-shaped pistillode, you can see here. And it contains many important horticultural groups. There are plenty of them growing out there. Adenidia virilii growing around the swimming pool here. Tychosperma, Wadietia, Veitchia, these are really, really important horticultural palms. You mess with the genera of these palms at your peril, because there will be a hate storm should you change the names. The anger from the horticultural community is not something you should wish upon yourself. However, thanks to the work of one of Sophie Nadeau's students, Elodia Alapetit, we have had to make some changes in the type of spermatine. This is part of her phylogeny, which firstly shows that we needed to sink the monotypic genus Sulphia into Balaka, which I won't dwell on here. At the same time, my colleague Charlie Diaspol in Papua, Indonesia, and myself were doing a lot of field work and turning up genera taxa, I should say, that we just could not place. They were clearly type of spermatine, but we could not justify their placement in any particular genus. And they ended up falling out in this position in the tree, where we would either have to describe them as separate monotypic genera, or lump them into pairs, or sink them into the whole damn lot here. And the least unpalatable solution was to describe them as separate genera, which is what we did. And they are rather distinctive things. Amazingly, they all come from a rather small area of Southeast Asia, from the head of the Bird's Head Peninsula in New Guinea into the Moluccas here. So there's one on Halmahera, one on Quindío, and one that's endemic to Biak. So these really are very local plants. And here is the Halmahera one, Gilaloa. We did describe it in Tychosperma, but that turned out to be wrong. A number of quite fine characters that I'm not going to get bogged down in. The spectacularly beautiful Manjekula with these arching, pendulous batwing leaflets from Biak. And finally, Wallaceodoxa. We had hoped to describe this in the centenary year of Alfred Russel Wallace. We made it to the 101st year, unfortunately, but this palm commemorates Alfred Russel Wallace. It means to the glory of Alfred Russel Wallace, the generic name, and it recognises the incredibly important work that Wallace did across the Milesian region, but also his interest in palms. And he did indeed visit Quindío, which is one of the sites where this palm occurs. Turning to sinkings, I've recently made a major change in the rattans. This was partly inspired by a paper published by Andrew Henderson and a student, Daniel Floder, sinking the genus Rhetus Batha, and in making that decision, they drew on some of their own unpublished data, but primarily on my own PhD research. When I did my research on rattan phylogenetics, I discovered that Calamus is seriously non-monophyletic, and the remaining 4 genera of Calamine are deeply nested in it. But after a long conversation and some soul searching, we decided to leave it as is. But I suppose the appearance of a paper sinking one of the genera changed my mind. To sink one of the genera is logically inconsistent. If you sink one, you have to sink them all. And so that's what I decided to do. And we now have, admittedly, a rather larger Calamus with 120 species added to make 500 species in total. I think this is a stable and pragmatic solution. It's not universally popular, but it can afford easier identification, especially when it's sterile. It would be nice to be able to do this on the basis of a better phylogenetic tree, and we're going to work towards that. But I suspect that even when we do have a better phylogenetic tree, there won't be a more appropriate solution. And just a heads up, here's a change that hasn't been made, but it seems very likely we're going to lose the genus Wallichia into Areca as well. So what about Hexapetion? This one is a bit controversial as well. Jean-Christophe, Pintaud and collaborators have resurrected Hexapetion, which is now understood, get the pointer, to be 2 species sister to the rest of Astracarium. And this is that split in the Bactridae phylogenetic tree of Roncal et al. So Hexapetion shares multiple characters with Astracarium, but it also has multiple distinguishing characters. One of the cases that could be made for making the split is that the split at the base of Bactridae is in fact one of the deepest splits anywhere in the Bactridae, so there's an element of phylogenetic distinctness. But of course, taxonomically, this is equivocal. We have chosen not to recognise it in genera Palmarum or the world checkers of plants, but there's an equal justification to use it if it suits your purposes. I think this case really underlines the fact that taxonomy is a matter of opinion, and it's a matter of choice. If you don't like the classifications of Astracarium, Hexapetion, or the ratans for that matter, it's really up to you to reject them, and time will tell whether the community will accept these taxonomic judgments. And just a quick plug for PalmWeb, which is a Palm biodiversity portal that we've been working on for many years now. All the genus level content, all the genus page content of genera Palmarum is in PalmWeb. If you don't have access to a copy of GP2, you do have access to the content, at least for the genus treatments, freely online through PalmWeb, so please make use of it. Okay. I'm now going to move on to phylogenetics. I love showing this slide because it's just the most astonishing palm. This is Hyphene compressor in Kenya, which happens to give me a lovely segue into, do I mean Hyphene coriacea? There's a sort of I forgot. I'm right, it's compressor. There's just a horrified look on Fred's face for a moment there. A nice segue into phylogenetic progress. Okay, so GP2 drew on 46 published studies, starting with the amazing prescient RFLP study that John and Natalie published in 1995 in the first conference proceedings, onto full-blown, large-scale sequence-based phylogenetics. Since 2009, so the year just after GP2, we've had about 17 papers published that contain novel phylogenies, new phylogenies generated for the purposes of the paper, which I feel is a bit surprisingly modest. I'll come back to that later. There have been very few densely sampled higher-level phylogenetic studies since GP2, although there have been some important studies at lower levels. These are really the key recent higher-level papers on plant systematics, so covering entire subfamilies or the family as a whole. Let's go back. This one came out well before GP2 and was the foundation for the circumscription of the subfamilies. These two came out after GP2, but I'll tell you a little secret. We really had the results before GP2, and they were part of the GP2 decision-making process. I just didn't get around to publishing until later. So we had the raw data. And I guess it's really this one in the middle that has been most important since GP2, because it provided a complete synthesis of available phylogenetic data sets. Something like 20 molecular data set, the morphological data set that John and Natalie developed over the years, and gave us the most highly resolved genus level, complete genus level treatment, phylogenetic treatment of the palms. And here you can see the 5 subfamilies mapped onto the complete genus level supertree, if you're not familiar with it already. And combined with a whole bunch of other resources that came to fruition around that time, we have found ourselves, as palm workers, all of us in fact, perhaps working with the largest tropical family with really complete fundamental biodiversity information tools. We have, as I just mentioned, a complete phylogeny. We have GP2, which is chock full of traits, if you care to mine for those traits. And we also have, for every single species of palm, distributional information, at least at a coarse scale, backed up by unrivaled specimen data. And combining that with publicly available large-scale data sets of ecology, edaphic features, climate, and so we are in a strong position to ask deep questions about tropical diversity. And that's what a lot of us have been doing. Many people in this room have been dabbling in this. And this led to Thomas Couvreur and I were asked to write a short opinion piece for BMC Biology, and we took the opportunity to, and we weren't the first to do it, frankly, but to make the case publicly for palms as a model group for tropical rainforest research. And we've been quite busy at that point, so we briefly synthesized several studies. So we've been able to use that super tree for historical biogeographic research, for looking at the phylogenetic structure of palm assemblages worldwide, to look at the correlates of particular growth forms, in this case the rattans, and even extending it to looking at the origins of rainforests as a whole. And this will be the topic of Thomas Couvreur's talk later. But putting those very high-level studies to one side, there have been some very important sets of work on particular clades. I want to highlight here again the work done by Christine Bacon that led to a set of 4 really nice papers. Here, first of all, dealing with the Trachycarpy as a whole, really resolving the phylogenetic relationships and putting a lot of concerns and confusion to bed, and also with a global biogeographic outlook. Another paper focused on the Panama Isthmus and the relationship between the American Trachycarpy and their relations into the taxa in the Pacific. A paper looking specifically at Tregear, which gave us all sorts of quite alarming insights into the definition of Tregear species. And finally, another paper that really focused on the Southeast Asian area and processes going on across Wallace's line. This is a really important kind of standard set of works now. Similarly, we've had a lot of efforts on tribe Cocosii. This has been much more pure, systematic phylogenetic, but important all the same. Several workers, some focusing on the tribe as a whole, some on several sub-tribes. And there'll be biogeographic analyses and some discussions around coconut origins. And really only one taxonomic change, which I have to admit until last night I'd actually forgotten in my slides, which is the sinking of the small genus Liticarium. Is robust, and that we don't need to meddle significantly with the higher level groups that we recognise in the palm classification. But there is still a lot of uncertainty, and I've got a screenshot here from the Supertree paper showing tribe Ariki, which is, that is phylogenetic uncertainty writ large there. We still have a lot of work to do to make things better. But I have had a sense, at least in what's coming out, and perhaps in the way that we're talking to each other, there's a bit of stagnation going on. And perhaps did GP2 satisfy our curiosity? Have all the low-hanging fruit been plucked so that people are prepared to engage in dealing with the incremental work that is required to really get a strong phylogeny? Or are we getting distracted on the bigger picture, the higher impact research of asking about the origins of rainforest? But things are happening. It's not quite as gloomy in practice as I have painted the picture just there. And I'd just like to highlight a few studies. We're going to hear from Soren Favio shortly. Soren has pulled together all available palm genetic data from GenBank, and he has intercalated it with all remaining species using various clever tricks. And we now have a complete species-level super tree of palms, which will be an invaluable tool for eco-informatic research. Going on at the same time is a similar study by Christine Bacon using the SuperSmart pipeline. She may be going to tell us a bit about this, I'm not sure. Are you going to tell us a bit about this later? A little bit about this later. And she's been generating new data and similarly pulling together lots and lots of data. One of the things I like about Soren's paper is the way he has mapped the phylogenetic uncertainty. On the right, I think, is my super tree, or one of the trees out of there. On the left is the results he gets, And only the black lines indicate a match in topology. So that's a really valuable thing. But another thing that's important to say about that study is that it's a critically important synthesis, but for the deep results, it's still effectively drawing on the same data we've been drawing on for years. And we really need to generate new data. It can't just be about synthesis. And there are people producing wonderful new data. We're going to hear from Craig Barrett on the last day. But Craig and his collaborators have been at the vanguard of embracing next generation sequencing techniques for palms. And here you can see a figure from a paper he has in review. I hope he doesn't mind me showing this, but I suspect he's going to show us all anyway. And the most important thing about this is that it strongly supports the GPU classification. And going on at the same time, a paper that has just come out by another one of Craig's collaborators in his broader collaborative network, using similar techniques. Jason Coleman has produced a phylogeny of the Aricoideae, which sheds interesting light on the deep relationships of the Aricoideae. So the most important thing I'd like to highlight here is the fact that Camidori are now strongly supported as sister to all the remaining Aricoids. And that is interesting because it was a curious thing that the Aricoideae, which has such an excellent, strong morphological synaptomorphy in the floral triad, had Camidori, which doesn't have floral triads. It has either solitary flowers or this amazing acervulous cluster, or anomalous triads derived from this. It was curious to see that nested within the Aricoideae, which was the topology that we accepted previously. In fact, I've even been wondering that if we had this topology when we were designing the new classification, would we have recognized a subfamily for Camidori, given that it violates the neat synaptomorphy of the triad for Aricoids. So these are really important new studies, although I would stress that the sampling, the taxon sampling, is still limited. But we have new opportunities, and these techniques are already being embraced. In Jim Lieben's Max lab in Georgia, Carolina Hayduk has been working on, has published recently a beautiful study focused on Sabal. I mean, quite narrow, I guess, taxonomically, but the important thing is the tool set that they have provided for us, which is the production of a set of tools by which we can reduce our sampling of the genome and derive a tractable set, but a very, very large set of DNA data from the nuclear genome using a hybrid baiting technique. We're never going to be able to sequence whole palm genomes and pull the data together, at least not in the short to medium term time scale. So having tools like that which can pull out a tractable set of data is absolutely essential for us to make progress. And this is now an invaluable tool for species level phylogenetics. But there's something weird about it too that worries me. It may be because I don't understand it properly. This paper contains a slightly disturbing figure. Let me explain this figure as best I can. So this is a histogram, and on the y-axis you have the number of gene trees, and on the bottom you have particular clades. So here we see that nearly 100, well over 100 gene trees strongly or weakly support the monophyly of Sabal. But as you go along here, you can see for the intergeneric relationships, the strong to weak conflict is dominating. So in fact, for the clades Sabal Bermudana plus United States Sabal, there is very, very weak agreement for that clade. So clearly, what this kind of approach is highlighting is something we kind of feared and knew already, which is that there will be lots and lots of disagreement between the individual gene trees that we derive using NGS methods, and we're going to have to find a way around that. So this is what I think is going on out there. There is a lot of work happening out there. Again, I take the risk that John took of offending by omission. I apologize if you or your research group aren't represented on this slide. I've used just one name from individual teams to try and get a fairly representative without making the slide too busy. There is, in fact, a lot going on. Even at the family level, I've just recruited a phylogenomics postdoc who will be pulling together data at the genus level across the entire family, and also looking at Tribariki. There's lots going on in other groups that I can't really list. If I missed you on here, please come and introduce yourself to me. I would love to hear about what you're doing. I gave the slide a bit like this. I'm nearly finished. I gave a slide like this at the IBC in 2011, where I tried to put our activities into a kind of taxon data space figure. Here we have a box that, well, I've just given away the surprise. Here we have a box that represents phylogenomic activity, lots and lots of data, but rather few species. The space that would normally be occupied by traditional Sanger sequencing driven phylogenetics, and then here a super tree, lots and lots of taxa, but perhaps holes in the data. And we need to find a way of getting into this sweet spot down here, where we have lots of data for all the species. I announced this idea, not such an innovative idea, at the IBC, and there was, there were tumbleweeds through the room. Nobody really took an interest. But I think we've reached a point now where this is already starting to happen. And that is very, very exciting. It's starting to happen because we have the tools to do it. And I think possibly the energy in our distributed consortium to do so. So we need to get into the sweet spot in the bottom right corner where we can build an all-species genomic level phylogeny. Why all species? Is it really that important? Do we need more data? Well, at a workshop recently, one of my colleagues said, a phylogeny is like a periodic table. And I thought that's just a fantastic analogy. No one would doubt the fundamental importance of a periodic table. There is no reason to doubt the fundamental importance of having a complete and accurate tree of life. And that is one of the reasons that applies. That's a reason that applies to all studies of life on Earth, but it's every bit as true for plants. We need a stronger framework for comparative plant biology, and an all-species phylogeny would give us an authoritative genomic resource, potential tools for other kinds of genomic applications. In particular, it would also provide a tool for barcoding. It's all right, you don't have to stand up. There's one more slide. You're looming and intimidating me now. Don't break my flow. Many other reasons to have a good phylogeny. We need a good phylogeny for predictive applied research, to explore, for example, palms as natural capital. Some of that's going on already with Rodrigo Camara-Leret's work, and perhaps also analysing extinction risks, which is something I'm quite keen to push further. And fundamentally to reinforce palms as a model group. And I put tractability at the problem. I think this is tractable. We really can do this. How will we do this? Well, this is kind of a vague slide. And it headlines just a few details. We have to mine our living and preserved collections. And I think it's the second one of those types of those collections that's most important. We need to find a way to consistently unlock our herbaria so that we can derive really good next generation sequencing data from it. We have to coordinate our field sampling. And we have to devise complementary protocols. They don't have to be the same, but we have to have complementary protocols that would mean that we could at some point come together and pool our data. And all of those things highlight the fact that cooperation is right at the center of this. We have to share our expertise. We have to share our data and our samples. This will give us a chance to go for consortium funding. And we have to find ways cooperatively to share credit and intellectual property fairly, especially to people early in their career stages who still need good papers to secure jobs. But in terms of IP, we also need to think about the CBD and the Nagoya Protocol, because a project like this will mean plumbing the depths of the genetic resources of numerous nations, and they have a right to care about it and feel credited for it as well. And finally, we have to be transparent. We have to share with each other our plans and our intentions. if we're going to remain as cohesive as the palm community always has been, especially in Europe, where we have a European network that meets annually. We've always been cohesive, but I get the sense of it not being quite so cohesive anymore. And I hope this meeting can redress that. So let's talk. Wolf Eisenhardt and I have been talking a lot about this at Kew lately. Wolf has recently secured a permanent position at Kew. Come and talk to us. We want to do this, but we want to do it together. Finally, some acknowledgements. Of course, once again, a tribute to John and Natalie, who have inspired me more than any other in my science endeavours. All my GP collaborators. Wolf, who is a constant source of inspiration and healthy argument, for which I'm extremely grateful. And I should say that having Wolf around at Kew has been invaluable during a very difficult year at Kew. Craig and Jason shared, and Soren shared their early results and let me show them. And Thomas, Daniel and Jens have also been part of a really wonderful collaboration over the last few years. And thanks to Lauren, not only for the talk title, but for being an unstinting support during her time as a botanist within the palm team. And thank you, Rodrigo, Gloria, and everybody else who have put together what I know is going to be an awe-inspiring and unbeatable, unfollowable palm conference. Thank you very much.